So we've talked about the different types of bacteria out there. Now I want to zoom in on the individual bacteria and look at their structures. Back in Bio 1, we talked about prokaryotic structures, and I think we all can agree there were 3 prokaryotic structures everything had. Plasma membrane, ribosomes, and the nucleoid region. But welcome to 102, where it gets a little bit harder, a little bit more complex, and it all comes down to that invagination we talked about in the last lecture. Broadly speaking, there are 2 types of cells. If you took my class last semester, you've seen this slide before. You've got bacteria, which are and archaeans, the prokaryotes, and you got eukaryotes, which is like everything else. And I really separated them out by lack of compartmentalization in prokaryotes, compartmentalization in eukaryotes. But it gets a little bit more complex than that. Everybody says prokaryotes have relatively simple structures. But the deal is, they are just as complex as we are. All prokaryotes, across the board, no matter where you look, archaeans or bacteria, are going to have a membrane. Now, that membrane may look slightly different, but they all have that phospholipid bilayer. They've got a polar head, nonpolar tails orienting in an aqueous solution so that you end up with a bilayer naturally forming. They all have what's known as a nucleoid region, a place where they keep their DNA. It's there to, it may be distributed through the cell, but it's there. You can find it. You might have ribosomes. You definitely have ribosomes. They're super important for creating polypeptides. No, was it two lectures ago where it's like, and let me tell you all about the differences in ribosomes. I'm not going to tell you again. There are a whole bunch of extra parts. Pili, flagella, the cell wall, and the glycocalyx. These are structures that some bacteria have, but not all of them. These structures, though, ultimately determined by their DNA, are going to provide advantages for the bacteria under certain conditions. Each of these prokaryotes has a certain shape to it, and that shape is given rise through an interaction between the membrane and the cytoskeleton. There's cocci, bacilli, vibrio, spirochetes, and spirilli. I know your lab only had 3. I expect you to know more. I will tell you on your exam, you're going to definitely have to look at a bacteria. Effectively, you're going to have to match the shape to the bacteria. I'm going to give you a bacteria's name, and you're going to have to describe what it looks like. The shape of the bacteria is determined by the intermediate filaments. Okay, back to 101. There are 3 components of the cytoskeleton. One of them is the intermediate filament. Do you guys know the other 2? I'll tell you, they're on this slide. Microtubules, microfilaments, otherwise known as actin filaments. So we've got the intermediate filaments provide rigidity and structure. So based on the way they interact with each other, you get a shape. One shape may be the coccus shape. Here you've got a single, coccus, which is just round, a sphere. Diplo, what does diplo mean? 2, so here you got diplococci, 2 spheres. Staphylo, you've got a pyramid shape. Strepto, a long line. But you'll notice that you've got that same sphere. Sphere, 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 sphere. Coccus are spheres. Bacillus are rods. If you have a streptobacillus, it's going to be a long chain of rods. Diplobacilli is 2. So it's the idea you got spheres, you got rods. Those are I think those are the ones we talked about in lab. Vibrios. Vibrios are comma-shaped. They look almost like elbow macaroni. Cheetos, maybe. I'm hungry. But they got that basic comma shape to them. Spirochetes are flexible spirals. They will, they're going to change shape under the scope as they move. Versus spirili, which are rigid spirals. They're going to move but maintain that same spiral shape. Difference between a cooked spaghetti and an uncooked spaghetti. I really am hungry. So those are your 5 different shapes that I'm going to want you guys to know. Spirili, Spirochetes, Vibrios, Bacillus, Coccus. And it will probably, like I said, be a matching section. Questions? People always say prokaryotic cells are not complex. We do get structures in prokaryotic cells based on the invagination of the membrane. So here we've got a basic prokaryote, and that membrane can invaginate by the movement of the actin filaments, the parts of the cytoskeleton, coming inward. So we've gone from having no compartments to having 3 distinct compartments. Different metabolic reactions can happen in each of these. Heck, you could maintain different pHs in those using buffers. This leads to a lot of interesting forms. The thylakoid. Where have you guys heard of thylakoids before? Not mitochondria, the other one. The chloroplast. We'll get to the mitochondria in a second. The chloroplast has thylakoid membranes. What a thylakoid is are flattened membranes that grow in from the cell membrane, and they have a lot of chlorophyll. So this particular bacteria will have an invagination, and another one, and another one, and another one, leading to these flattened discs, like that. Why are there twists and turns here? What does that do? When in doubt, surface area. It creates a lot of surface area. So the more surface area there is, the more chlorophyll this can hold. The more chlorophyll it can hold, the more sugar it can make. Again, we talked about endosymbiosis. Here we've got this thing with a whole bunch of chlorophyll in it. If it was to be eaten by a larger cell, but not digested, now you have a double membrane-bound organelle with its own unique set of DNA. Magnetosomes form by invagination of the membrane. Here, just like with the thylakoid, you've got these invaginations. What occurs is a small bead of, here, so we've got our cell, you got invaginating, sort of like this. And we'll just do it like that. So you end up having these little compartments inside. In each of those compartments, a little crystal of iron can grow. Iron is magnetic, thus magnetosome. These iron deposits inside the cells allow the cell to have an idea of north and south. It gives them directionality. There's some research that shows birds have a lot of magnetosomes in their beaks. So what does that let them do? Figure out where the Earth's magnetic field is aligned, allowing for migration. Obviously, bacteria don't do terribly much migrating, but it's useful for them to determine what direction to move in, especially if the direction is toward iron or a source of food. Some bacteria have nucleus-like bodies, not true nuclei, but they have a nucleoid region that's surrounded by invaginations. Once again, we're getting close to, let's talk about possible beginnings of eukaryotic life. This may be how the nucleus originally formed. It is a we don't know for sure. But if you have these invaginations coming in like that, you've got sections acting as nuclear pores. You've got your DNA fully protected. So some bacteria have that now. Outside the bacterial cell, you might have what's known as glycocalyx. Glycocalyx is super awesome, and I didn't realize until I wrote all the lectures for this course how ubiquitous it is, how everywhere it is. It is thick and slimy. It's made up of polysaccharides, proteins, and lipids, and it surrounds bacterial cells. It acts almost like a shield. So you've got your phospholipid bilayer, and then outside of that, a layer of glycocalyx. I do get way too excited about glycocalyx. I will definitely preface it. I am so super enthusiastic about glycocalyx, and it's going to come up a lot. Here's what it does. I'm going to go through each of these functions. It holds cells together. It helps bacteria float in water, repels attacks, binds nutrients, and protects the cell from ultraviolet radiation. So first off, hold cells together. Glycocalyx acts like glue, literal glue. It's cement. It will bind them not just to each other, but to a surface. If two cells are bound together through their glycocalyx, they can easily pass DNA back and forth. They act as a unit. Think of it as like this slimy substance that just utterly surrounds organisms. It provides a okay, have you ever made Jell-O that had fruit in it? Okay, the individual fruit in it would be the bacteria. The glycocalyx is the actual Jell-O. So everything is held in place. It makes things act like a unit. Glycocalyx helps some bacteria float. Literally, bacteria will take this slimy stuff around them and start to excrete gases into it to create bubbles. It acts like water wings. You remember water wings when you were younger? They're the things that like you put on, it made you look like you had muscles. Bacteria use those, so they float without using any energy. They don't need to use flagella or anything like that. You just add more gas, take gas away. It helps them repel attack. It acts like a shield. The first defense your body has when you eat a foodborne pathogen, you eat some sort of bacteria in your food. I don't know, you had a bad egg sandwich. I don't know. Got it out of a vending machine because that's crazy. You eat it. This bacteria that was growing in it goes immediately to your stomach. And what does your stomach do? Breaks it down because you got acids in there, right? The proteins can't handle it. What glycocalyx does for that bacteria is it provides a shield. So the acid has to eat away at that shield first. And if the bacteria's glycocalyx layer is thick enough, it can survive in your stomach just long enough to get passed into your small intestine, where the acid is neutralized by buffers. And then once it's in there, then it can start reproducing, and you get messed up. It binds nutrients. This is odd to think about, but you guys at some point in your life have been chewing gum and it magically fell out of your mouth. You don't know how it did. There have been times, I swear we've all had this experience. You were chewing gum and then it's not in your mouth anymore. Maybe you went to talk, maybe you sneezed, I don't know. It's on the floor. Do you pick up that gum and put it back in your mouth? No. Why? What's all over the gum now? The ground, germs, sure, but there's stuff on. You pick it up and it's literally covered in stuff. It's like when you drop 5-second rule, right? It's when you drop a lollipop that you've been sucking on. It picks up all this stuff. There's stuff then on it. Bacteria use sort of that same concept to their advantage. They've got this super sticky, gross layer on the outside. So as they're going through their environment, stuff lands on it. Well, the bacteria is like, I'm going to eat that now. It acts almost like a big spherical net that the bacteria can use in order to grab food. And then protects from ultraviolet radiation. Ultraviolet radiation is dangerous to bacteria, very dangerous. Ultraviolet radiation will lead to damage to the DNA. And if the DNA is damaged, it can't be faithfully replicated, the bacteria dies. Now here's so if you're sick, one thing that people say to do is make sure all the windows are open. The blinds are open, because ultraviolet radiation from the sun kills bacteria. If you're in a lab, you usually have a UV light that you put over your area. Why? To kill all the bacteria. Bacteria do not suffer ultraviolet radiation unless they have a thick shield of glycocalyx. That glycocalyx actually provides sunscreen, blocking that radiation so it doesn't impact the DNA. So glycocalyx does a whole lot. But probably to me, the most interesting thing that glycocalyx does is it produces what are known as biofilms. Glycocalyx will stick to a surface, like glue, holding the bacteria in place. And then other bacteria cling to that first bacteria, and you get a mound building up. So this produces a favorable environment. If you've ever been if you've ever played in a creek, I don't know if you guys had a childhood. If you played in a creek, you'll notice that there's this really slippery green stuff growing on rocks. Right, which is, it is held in place by glycocalyx. Building up on, it's very, very tough to break off. So glycocalyx holds bacteria in place where they'd otherwise wash away. Think about your mouth. Don't think too hard about it. It's really gross. Your mouth is seriously gross. Inside your mouth, you've got bacteria that want to grow on your teeth. We agree on that, right? You've been to the dentist. They're held in place by glycocalyx. You swallow an awful lot of saliva a day. You swallow, and we'll talk about this later, about 1.5 liters. So think about a wine bottle's worth of saliva a day. All of that is trying to move bacteria. I'm not trying to move bacteria, but the bacteria have to survive in your mouth while holding on while you've got all this liquid rushing past. They do it through glycocalyx. Bacteria are also going to use this glycocalyx for what's known as quorum sensing. Okay, now we're getting a little bit more complex. Into this biofilm, you get a population of bacteria in this glycocalyx matrix. Each of them secrete an individual molecule. That's a communication molecule. It says, I'm ready. The bacteria doesn't do anything until there is a sufficiently high amount of that molecule that says, I'm ready, before it does anything. So you've got a population that's building up. They're each putting out molecules that say, I'm ready to do X or I'm ready to do Y. When the glycocalyx gets filled up with enough of a single type of communication molecule, suddenly every cell in that group will do that same process, whatever that process may be. You've all, at some point in your lives, gotten sick. I know, because I've gotten the notes. When you get sick, we all, and you've probably recognized it at this point, you all can feel it coming on, right? You're like, I'm going to be sick in like 2 hours. I better get all my work done now. Do you guys know what I'm talking about, that feeling? The bacteria that are getting you sick have been in your system for days at this point, but they haven't done anything aside from reproduce, get bigger. They didn't enter into this pathogenic phase until this level of that signal molecule builds up in the glycocalyx matrix around them. Once you get a high enough level, suddenly they all say, okay, now we're ready to attack. Now we're ready to make this other thing happen. Does that make sense? And then suddenly, all the bacteria switch into a different mode. They switch into a different way of interacting with the world. That's quorum sensing. shoot, there's this whole new field of microbiology. What's it called? It's going to kill me, and I'm going to totally forget it. All right, so let me describe it to you. You'll notice that when you're sick, you feel depressed. You feel sad. You're like, and you become a big baby. I mean, let's be honest. You become a big baby when you're sick. Your emotions cause a chemical signal to be delivered to your gut. It's connected, right? If you are depressed, it will cause the bacteria in your gut to grow faster. So therefore, so if you're depressed, you tend to get sick more often. The other way works as well, and this is what's blowing all these scientists' minds right now. If you've got bacteria growing in your stomach, or not your stomach, in your intestines, and they're producing this chemical, it will feed back to your brain and make you get more depressed, which is what causes those emotional changes. Turns out that if you act happy, this is so weird, if you act happy, if you produce the same chemicals that would happen when your brain is happy or excited or aroused, that sends a signal down to your gut that prevents the bacteria from reproducing. It is so freaking weird and awesome. I've got to find a TED Talk for you guys on it. Yes?
If I have to guess, it probably has patho in it.
It probably does have patho.
Considering it relates to emotions and pathology.
It could. It's got something more to do with the microenvironment, so it's going to have microbiome in it. But super interesting field. If any of you guys are going into medicine, it's sort of cutting edge right now. It's building up in the research behind it's building up. This all leads me to a discussion of why you should brush your teeth. Now, you probably all know it's important to brush your teeth, and probably you do it for two reasons. One, you don't want to have sort of stains in your teeth, and two, you want to be able to talk to people without embarrassing yourself. Dental plaque are bacteria that are holding on to your teeth using glycocalyx. Man, I'm really harping this glycocalyx thing. They're holding on to your teeth using glycocalyx. They're cementing themselves in place. So they're stopped from moving when you take a fluid in or when you're using saliva. The only way to get rid of it is through mechanical action, which is why you brush your teeth. You'll notice that no matter how much mouthwash you use, you're not removing plaque. You have to take a toothbrush, or our ancestors would take a stick. Not kidding, that's how we brushed our teeth. Stick to the teeth. And then you would apply some sort of cleaning agent. That's why you use a toothbrush and toothpaste. It's the only way to get plaque off. The deal is, though, that only works if you do it within 24 hours of the glycocalyx being laid down. After 24 hours-ish, depending on the bacterial strain, that it's not just glycocalyx holding it in place. It's glycocalyx plus some of the calcium carbonate that from the teeth. So then you've got more of a what's it called? More of a permanent problem. I guess semi-permanent. It's more of a, instead of dealing with glycocalyx as glue, you're dealing more with cement. In order to get that off, you have to go to the dentist, and they take that little hook thing and they scrape away at that. So brush your teeth at least once a day, preferably twice a day. Life good? All right. Beneath that glycocalyx, the bacteria will have cell walls. Cell walls are there for a few reasons. One of the most important is to prevent osmotic lysing, or prevent the cell from exploding. Sort of, it serves the same purpose as it does in plant cells, but they are made of entirely different macromolecules. It's there to stop the cell from lysing in hypotonic conditions. Not sure if you guys remember, we talked about tonicity in Bio 101, hypotonic, hypertonic, isotonic. It's isotonic, you're going to get water moving in and out at the same rate. If it's hypertonic, water leaves the cell. And if the solution is hypotonic, water floods into the cell. Now, if there's not something in a hypotonic solution, pure water, that stops the cell from continually expanding, it will pop. The wall in plants and the wall in cells and bacteria stop it from popping. Most of the cell walls in bacteria are made up of peptidoglycan. So this is carbohydrates that are cross-linked. Just like we talked about with phospholipids, these walls have extra supports in them, holding them together. And because of that, that leads to our two big bacterial types, the gram-positive and the gram-negative. So I want you guys to understand differences between these two. Two big types, gram-positive, gram-negative. And it's all based on the thickness and composition of the cell wall. First off, we have our gram-positive bacteria. They have a very thick peptidoglycan layer. So it's sort of, you've got plasma membrane, and then this exposed peptidoglycan layer. They're gram-positive because the stain stains the peptidoglycan. These are also going to be the bacteria that are going to be most susceptible to antibiotics, because the antibiotic can get through this layer and one plasma membrane layer. So the gram-positive have a very thick peptidoglycan layer. The gram-negative, on the other hand, have a thin peptidoglycan layer, but a double phospholipid bilayer. So you got phospholipid bilayer, peptidoglycan layer, phospholipid bilayer. And back to me being hungry, the way I remember this, I kid you not, the way I remember this is I think about pies. The gram-positive, so positive starts with a P, right? So this is so stupid. Gram-positive, I think of pumpkin pies, which also starts with the letter P. Because pumpkin pies have a layer of crust, and then they're just filled up with the pumpkin. Does that make sense? So I'm thinking that's the peptidoglycan layer, and then you have the membrane. That, of course, is compared to the gram-negative, which is, the way I think of it's like an Oreo cookie. You've got a layer of phospholipid bilayer, a layer of peptidoglycan, and a layer of phospholipid bilayer. That's the way I remember it. The gram-positives are going to be susceptible to bacteria. The gram-negatives are not. I'm sorry, are going to be susceptible to antibiotics. Gram staining is a process that does actually, we could do it in the lab. Really easy. It's just a question of staining with one color, staining with another color, drying them off with a hairdryer, looking at them under an immersion scope. It's not a difficult task. What is difficult to do is to do this without getting stain all over your fingers. And I'm like, yeah. So gram-positive susceptible to antibiotics, gram-negative not, which is why when doctors go to prescribe antibiotics, they're going to test to see whether positive or negative. If it's a gram-negative bacteria, they're not going to give you an antibiotic because it's just not going to work. There are so many antibiotics out there in the environment right now, doctors are actually very conscious of only subscribe them when they will actually be efficient rather than subscribe them so the patient feels better and gets out of the office. Questions? Bacteria's got to move. They got to get from point A to point B, and they can do that using several mechanisms. They can use flagellar pili. We talked about that a bit in Bio 101. If you're going to use a flagella, or flagellome, you're going to pretty much have a rotor with a whip on the end. That rotor has motor proteins that walk along a filament, grabbing and spinning. It reminds me of, oh, what are those things called? Again, back to your non-existent childhoods. Did you guys play on a playground when you were younger? Yes, okay, we got the playground idea. With you guys, when you were younger, did you have the playground equipment that is now pretty much universally banned? It was a big, round merry-go-round thing. And you get in, and like you'd have like the slow people sit in the middle, and the really fast people on the outside running as fast as they can, until eventually they got to a point where they just like grab and throw and grab and throw. You guys know what I'm talking about? Okay. What's doing the grabbing and throwing are the motor proteins sitting right around that intermediate filament. They grab and they pull toward them, and they release using energy. Then they're going to grab, pull, and release. And as that happens, this tip starts to spin. And it spins faster and faster in one direction. Now, attached to that rotor, you have a long filament. The longer the filament, the better. So if I have this and I'm moving it side to side, it's barely perceptible, you know? But if I extend this ruler out longer and I make the same movement, suddenly the tip is moving farther and farther. Does that make sense? So if it's even longer, you're going to have a huge movement based on a little action. So now you've got this rotor spinning like mad, so it pushes it forward in one direction, or it spins in the other direction and it pulls it back. It's like a little fishing boat motor. The cell may project pili. Pili are not cilia. I do want to make that very clear. Pili are not cilia. Pili could move through an extension and pulling. They're more like, think about tentacles, reaching out, grabbing, and pulling. Reaching out, grabbing, and pulling. So they can pull themselves through their environment. Questions on those two?